Hi, everybody. Good to see you all again. I feel like I only see you once a year around this time. But I thought I would start with a little trip down memory lane. If it would go forward. Oh, there we go. Okay, so spruce budworm, native pest, been here a long time. If we look back in the records, we really started keeping good records in the early 1900s. The first outbreak that there's a lot of information from is the 1910s. And this one was bad. It was very severe. It covered a good chunk of the state. And at the time, there wasn't really forest management. It was kind of treated like wildfires. You just let it burn out. And that's exactly what they did with the first spruce budworm outbreak. And so overall, it ended up killing 27.5 million cords of spruce and fir. And then we fast forward 30-ish years, which is the normal time for spruce budworm to act back up. And we had the outbreak in the 1940s and 50s. And this one is considered relatively mild. There are some locally intense areas. So we do see some regions on the Quebec border and in northern Maine. At this time, they started thinking about management, thinning stands. DDT was being tested as well during this time. But it did not do what the 1910s outbreak did. And then we fast forward another 30 years and we get to the point that some might remember. And this is the really big outbreak again in the 70s and 80s. It was also very severe. This was the era of aerial spraying. So over 10 million acres were sprayed during this outbreak. And 20 to 25 million cords of spruce and fir killed. So kind of comparable to the 1910s outbreak. Fast forward 30 years again, and we are where we are now with the spruce budworm outbreak starting up in Canada. So kind of as a refresher, I was started here in 2020, and in October 2021, I opened the Spruce Budworm Processing Lab here at UMaine under the sort of it's coming mentality, and we need to be prepared. So the first year we had about 300 monitoring sites across the state, and we're looking for that magical 7 L2s as the threshold. And we found 2 locations. One was up near Cross Lake, and the other was on the Quebec border. We then did some extra sampling at these locations. So the landowners for those properties sampled more to kind of get an idea of what might be the high risk zone. So 69 additional sites based on like expanding a radius around the hotspot, and 3 more were found. Well, 3 hotspots total next to the close ones. An average, the highest average we found was 23.67 L2s for a single site. But those were treated, which were good. And then in 2022, we sampled, again, around 300 monitoring sites. None of them were even close to 7 L2s. There was no additional sampling done, and there was actually quite the decline across the state. And I assume perhaps the Canadians are going to speak of this as well, but I think they saw a decline in the L2 numbers in 2022 as well. So everything was looking kind of good until this year. We expanded the monitoring program. We received federal funding. We leveraged CFRUs and got additional funding to expand the monitoring program and lower the fee for service costs. So we did 324 sites, and we found 2 hotspots. So the first one was right outside of Fort Kent. It's my understanding it was a pretty small woodlot. And then the other one is again up on the Quebec border, less than 10 miles from the 2021 hotspot that was found. This has resulted in an extra 54 samples being collected for that area. So far we have now found 22 hotspots, and they are all in this zone here. The highest we found was 35 L2s for a single site. The location up in Fort Kent didn't do any additional sampling. So if we zoom in on this particular area on the Quebec border, basically anything, it's kind of hard to see the colors, but anything yellow, orange, or red is above 7 L2s, or anything kind of purple down here. So if I were to connect the dots, which I attempted to do, you get a shape that kind of looks like that, which is about 20 miles long of the Quebec border. But Neil's way better at this than me. And so he was able to take the data and run it in the model and kind of determine what is the general area of concern. And so this is a very rough estimate, he told me to tell you. And it's about 54,000 acres. But within there, he also tried to remove like the hardwood areas and the areas that have had spruce or fir removed. So it's about 37,000 acres. And even though these two townships, these should also kind of be on the orangish, yellowish, we were unable to get kind of enough samples, and now it's the mud season. So we probably won't be able to tease that apart, but it did go down into further down into these two townships. And in fact, we found one that was like right on the border of even this township. So unfortunately, our 2023 estimates are probably an underestimate, just based on the timing. And now it's a little late in the season to get any more additional samples before they wake up for the summer. So this area kind of corresponds to a lot. It corresponds to, sorry, Mike, I did have one little spoiler I threw in here. So the pheromone catches we see, and Mike will probably show the same slide, that it corresponds to the area where we were finding high L2s, and almost like on both sides of our zone of where we were finding things. This was in 2022. It was a high catch count for adults in the pheromone traps as well. So there does seem to be some correlation that we should be looking at areas that have high pheromone trap counts. The other thing is this corresponds to where we have some of the densest spruce fir forest. So again, we're looking in this region here. And if you see the dots on here, these are part of the 300 sites in the state that we do for the monitoring program. And unfortunately, you will see kind of some big gaps that we do have. Unfortunately, some of the densest area actually isn't currently part of the monitoring program, which is something to discuss for future. It also corresponds to Quebec's infestation. So this is 2022's projections based on their L2 counts. And you can see that the highest zone is almost right over the border where we started finding higher L2s in 2023. So all of this kind of lines up that this zone of the state is pretty much our high risk zone now. If we look at just all of the L2 data, this is now my third year of the L2 processing lab. So we're able to kind of look at some changes and how are things going. So in 2022, you know, this is all 300 sites average. We don't see too much change, 0.28, 0.39, 0.29, you know, a little bit, but we had a tier system where the high risk sites in the northern part of the state were tier 1, and then tier 2 moving down to tier 4 as we considered low risk. And you do see in tier 1, besides the weird 2022 drop in L2s, there's definitely an upward projection in overall L2s at those high risk sites. If we look at just like, what are these sites doing? Are they increasing or decreasing or staying the same? Again, this is all 300 sites. So we do see the majority of the sites are just staying the same. Those that are increasing, so anything in the red, orange, yellow is increasing, anything in blue is decreasing. We see most of the growth, again, is in the northern part of the state. Balsam woolly adelgid. So the spruce budworm processing uses hot sodium hydroxide to dissolve the silk. It turns out it also dissolves the wool that woolly adelgids make. And so we started realizing that we can actually do some balsam woolly adelgid counts for the branches that come in. And so this is our second year of doing those counts. And so we are seeing that it's actually the middle part of the state. We have some of the highest balsam woolly adelgid counts. However, it is still unknown what the relationship is. We know that it's out there, it is invasive, it can be detrimental in high densities, but we don't know whether or not the counts that we're getting constitute being high density or not. So in the future, I'm hoping to bring on a grad student possibly next year to kind of look at this relationship between spruce budworm, balsam woolly adelgid, and overall tree health to kind of see what the relationship is and to see if we can possibly develop some threshold analyses for these balsam woolly adelgid counts. Overall, we did find them in more sites this year, but that's not necessarily anything until I learn more about its life cycle in Maine. It could just be when the branches were clipped. They were during the crawler stage this year compared to last year. Maybe they weren't in the crawling stage. So we are finding it. We are giving you all the numbers, but we don't actually know what it means quite yet. So this is something I hope to do moving forward. Speaking of moving forward, so this year has been very informative, kind of starting to scare us a little bit. And so we want to sort of think about what we're doing and how can we improve. So on our end, some of the things we want to do is update the tier system. So this was based on 2020's data, which the hotspot found in 2020 was over here, which is why these weren't listed as tier 1 sites on the Quebec border, because we hadn't really caught much over there. Historically or otherwise. And so we definitely need to redo the tier system to make sure that the Quebec border here is actually going to be a new tier 1. So those would be the first samples that are processed when they come in. We definitely want to add more sites if possible, based on, of course, landowners, but you'll see we are missing any monitoring sites. There's none in this entire zone. Which is right where we think if there is high numbers, it's going to be in that area. So we do want to try to get more plots out there. More frequent deliveries from Fort Kent. Most of the samples clipped up here go to Neil up in Fort Kent, and Neil has been amazing, made multiple trips to Orono or sent with folks that were heading our way. But one of the reasons why we don't know the size of this current hotspot area is because the hotspot that was found wasn't clipped until the end of November. And then because of the holidays and everything, it wasn't delivered to Orono until after New Year's. And then because it was listed as a tier 3, it wasn't processed until the end of January. And so we really want to try and make sure that we get those samples much sooner than the end of January. As far as from you guys, if possible, try to clip branches sooner, especially from these high-risk zones, either in this area, which is still a concern area, or the Quebec border area, to get those as soon as we can. The lab is up and running and ready to start processing, usually by late September, early October. You can start bringing stuff in. Collect from more locations. If you are one of the landowners that has property up there, we can do 350 monitoring sites. We did not reach that this year from just the monitoring program. So we do have wiggle room to add another, I think, 20 or 25 plots, and maybe we can ensure that they're on that region. And then at the meeting later, you guys are going to be asked a question, which is, would you allow the Spruce Budworm Lab to notify neighboring landowners if we find a hotspot on your property? So if it's within like 5 miles, 10 miles, whatever we agree to, that we can notify the other landowners. That was another thing that we were kind of tripping over a little bit, like, wait, can I let so-and-so know that maybe they should go back out and recollect in this area based on what we found nearby? So I just want to make sure that we're all on the same page, that everybody is okay, or if you're not okay, that we know that before kind of tackling this next upcoming season. So I will just end really quick, you know, in looking historically at spruce budworm, it has been variable. It's not always, if Canada's experiencing a severe outbreak, that Maine does. And so we don't quite know what is going to be happening. Could be a little bit of everything, but we do need to make sure that we continue to monitor in order to figure out what the populations are doing in the state. So I'll end there. I will mention that my lab manager, James, has moved on and found a permanent position. I'm very happy for him. He's now working for the NRCS office in Bangor. But so at the moment, you're stuck talking to me if you have any issues with the lab, and I'll be looking to rehire or hire a lab manager to run this upcoming season's processing. So hopefully that'll be done over the summer, and I'll let you guys know when that's done. I don't know if I have any time for questions though.
Can I jump in from Fort Kent for a second?
Hi, Neil.
Hello, Angela. Hello, everyone. So I just want to make sure I'm not taking too much credit for that map there, that I started my own interpolation models at the same time as I sent a request to the Canadian team, and the Canadian team delivered the through their interpolation model at the same time I was finishing mine. So I get to pat myself on the back, so I did a very good job, but I ended up using theirs, which is, I think, a more reliable model.
I just have a question about redoing the tier system. That's something that your lab will model and then
Yes.
basically just let us know.
Yes.
Okay.
Yeah, we'll redo that and let landowners know. And then the other thing Neil and I, it was this morning, talked about is that maybe if we get Mike's pheromone data, depending upon when that's available, that we might use that to also develop some of the tiers. Yeah.
I think just going off that, Angela, that might be a good discussion topic as well. We kind of run this program, it's a lot of work to get to these sampling sites. A lot of the L2 sites and the pheromone sites are co-located. So we have people do them at the same time to save a lot of, hours basically. But if people have an appetite for getting that data sooner, the moss samples can certainly come in sooner than the L2s. L2s are kind of hemmed in by the weather and sampling when it gets a little cooler, but yeah, especially in a year like this where we see this, we could definitely try to count certain localities way sooner than the L2s come in and kind of have some of those predictions.
All right, thanks.